Hello everyone, today we are going to talk about one of the most important topics in biochemistry, which is amino acids. Content which is covered in our research project is definition of amino acids, properties of amino acids, structure of amino acids, classification of amino acids, and functions of amino acids. The first one is definition of amino acids. Amino acids are molecules containing an amine group, a carboxylic acid group, and a side chain that is specific to each amino acid. The key elements of an amino acid are carbon, hydrogen, oxygen, and nitrogen. These serve as building blocks of proteins. The second thing is properties of amino acids. Properties are physical properties and chemical properties. Physical properties of amino acids. Amino acids are colorless and crystalline solid, and they have a high melting point greater than 200. And when talking about solubility, they are soluble in water, slightly soluble in alcohol, and dissolve with difficulty in methanol, ethanol, and propanol. And on heating to high temperatures, amino acids decompose. And also amino acids except glycine are optically active, and they are responsible of peptide bond formation. Chemical properties are zwitterionic property, amphoteric property, non-hadron test, Zansobrotic test, reaction with Sanger's reagent, reaction with nitrous acid. The first one is zwitterionic property. A zwitterion is a molecule with function groups of which at least one has a positive and one has a negative electrical charge. The net charge of the entire molecule is zero. Amino acids are the best known examples of zwitterions as they contain an amino basic group and a carboxylic acid group. The amino basic group is a stronger base, and so it picks up hydrogen ion from amino acids exist in solution. And here we can see that at low pH or acidic medium, amino acids act as base, and at high pH or at alkaline media, amino acids act as acid. And at neutral pH, amino acids act as zwitterion. The second one is amphoteric property. Amino acids are amphoteric in nature, that is, they act as both acids and base since due to the two amine and carboxylic group present. The third one is non-hadron test. When 1 milliliter of non-hadron solution is added to 1 milliliter protein solution and heated, the formation of a violet color indicates the presence of alpha amino acids. The first one is Zansobrotic test. It's performed for the detection of aromatic amino acids like tyrosine, tryptophan, and phenylalanine in a protein solution. The nitration of benzoyl radicals present in the amino acid chain occurs due to reaction with nitric acid, giving the solution yellow coloration.
Structure of amino acids. All the 20 common amino acids are alpha amino acids. They contain a carboxylic group, an amino group, and a side chain R group, all attached to the alpha carbon. As we see here, the carbon atom is attached to carboxylic group, amino group, side chain, and hydrogen atom. Exceptions are number 1, glycine, which doesn't have a side chain, its alpha carbon contains 2 hydrogens. Number 2, berylline, in which the nitrogen is part of a ring. Number 3, thus each amino acid has an amine group at one end and an acid group at the other and a distinctive side chain. The backbone is the same for all amino acids, while the side chain differs from one amino acid to the next. Number 4, all of 20 amino acids except glycine are of L configuration as for all, but one amino acid, the alpha carbon, is a symmetric carbon because glycine doesn't contain a symmetric carbon atom. It isn't optically active and thus it's neither dextro nor levo. In this slide we will talk about classification of amino acids according to side chain. Non-polar, uncharged polar, acidic, and basic. As we see here, examples of nonpolar amino acids: glycine, alanine, valine, leucine, isoleucine, beryline, phenylalanine, tryptophan, and methionine. Examples of uncharged polar amino acids: glycine, serine, asparagine, glutamine, threonine, cysteine, tyrosine. In here, acidic amino acids like aspartic acid, glutamic acid. And finally, examples of basic amino acids: lysine, arginine, histidine.
Classification of amino acids based on nutrition: essential amino acids, semi-essential amino acids, non-essential amino acids. Essential amino acids can't be synthesized in the body, should be taken in diet, like valine, tryptophan, phenylalanine, lysine, methionine, threonine, leucine, isoleucine, arginine, histidine. Semi-essential amino acids can be synthesized in the body, but the rate of synthesis is lesser than requirement, needed during growth, pregnancy, or repair, like arginine and histidine. Non-essential amino acids can be synthesized in the body, thus their absence in diet doesn't adversely affect the growth, like serine, alanine, proline, hydroxyproline, glycine, tyrosine, cysteine, cysteine, aspartic, glutamic.
Classification of amino acids based on metabolic fat: first glucogenic, second ketogenic, third glucogenic and ketogenic. First, glucogenic amino acids give glucose inside the body like glycine, alanine, aspartic and glutamic acid. Second, ketogenic amino acids give ketone bodies as leucine. Third, glucogenic and ketogenic amino acids. They give both glucose and ketone bodies like lysine, isoleucine, phenylalanine, tryptophan, and tyrosine. Functions of amino acids: 1. In particular, 20 very important amino acids are crucial for life as they contain peptides and proteins and are known to be the building blocks for all living things. 2. The linear sequence of amino acids residues in a polypeptide chain determines the three-dimensional configuration of a protein, and the structure of a protein determines its function. 3. Amino acids are imperative for sustaining the health of the human body. They largely promote the production of hormones, structure of muscles, human nervous system's healthy function, the health of vital organs, normal cellular structure. 4. The amino acids are used by various tissues to synthesize protein and to produce nitrogen containing compounds like purines, heme, creatine, epinephrine, or they are oxidized to produce energy. 5. The breakdown of both dietary and tissue proteins yield nitrogen-containing substrates and carbon skeletons. 6. The nitrogen-containing substrates are used in the biosynthesis of purines, pyrimidines, neurotransmitters, hormones, porphyrins, and non-essential amino acids. 7. The carbon skeletons are used as a fuel source in the citric acid cycle, used for gluconeogenesis or used in fatty acid synthesis. Thank you.